All right, I think we're ready to get started. Thanks, everyone. I'm Joshua Schimmer from the Piper Jaffray biotech team. No doubt in my mind that this is the most important session of the day because, well, first of all, it reflects the progress we've seen in gene therapy, but it also reflects really the need to take what we've seen and really optimize it for broad-based human therapeutics to create really a robust platform, a very modular platform where you know, that the mountain of disease with unmet need of a genetic basis can really be tapped into and addressed. So I think what we'll do is get each one of our panelists to give a quick intro and snapshot of their companies, and then we'll talk a little bit about what they're doing to optimize gene therapy and gene edit delivery. So David, why don't we start with you?
Great. Thanks, Josh, for having me on the panel. It's great to be here. I'm Dave Kirn. I'm the CEO and co-founder of 4D Molecular Therapeutics. And briefly, at 4D, we're a gene therapy company with a robust discovery platform that allows us to identify highly optimized and proprietary AAV vectors that can be targeted to any organ or specific tissue in the human body. And we expect that this will allow us to unlock the full potential of gene therapy by through the use of optimized vectors. Our vectors are generally designed to have three key features. Number one is they're highly targeted and efficient at targeting specific tissues or even cell types within specific tissues. Number one. Number two, they're designed to be delivered by routine and optimal clinical routes of administration. And then number three is they can be engineered for evasion of pre-existing antibodies in the population, which can be a detriment to some of the current AAV vectors. Our pipeline, we have our own internal programs going after monogenic recessive diseases with high unmet medical need, including choroideremia with intravitreal injection for the retina, and then cystic fibrosis with a vector that's optimized for aerosolized delivery. And then we also have a robust partnership program. We have 5 partnerships currently, including Pfizer, Roche, Unicure, and AGT, as well as Benetech. So thanks, Josh.
Great. John?
John Leonard. I'm the Chief Medical Officer of Intellia Therapeutics based in Cambridge, Mass. Newly formed about a year and a half ago, and we're a CRISPR-Cas9 gene editing company. Not as far along as some of the other companies here on the platform, but moving very quickly in a very exciting space. The entire notion of the company is to have very specific changes in situ to essentially modify any gene, anyhow, anywhere. And currently we're focused primarily on in vivo targets, especially focusing on the liver. And many of you will know that we struck a relationship with Novartis as a key partnership some months ago.
Great. Steve?
I'm Steve Paul, the CEO and co-founder of Voyager Therapeutics. We work in R&D in really 2 dimensions. One, we have what we call a product engine or a platform, so we're focused on vector engineering, capsid engineering, delivery, which we think is essential for success of any of these programs, and manufacturing. We've invested in a manufacturing process that involves baculovirus SF9 cells, which we believe is a high quality and scalable process. On the other end, we are very focused on our product engine, our programs, I should say, our pipeline. We have a program in advanced Parkinson's disease, which is currently enrolling in a phase 1B dose escalating, dose ranging study. We are dosing our second cohort of subjects with some encouraging clinical data in the first patient dosed in the higher dose cohort. This is a program that delivers a therapeutic protein, a gene that encodes an enzyme that allows for the conversion of levodopa, a medicine, to dopamine right in the area of the brain that's depleted of dopamine to get the 100,000 or so patients who are refractive, if you will, to levodopa therapy back to responding very well. The other programs are preclinical programs, one for a monogenic form of ALS, SOD1 mutant ALS, knockdown strategy, one for Friedreich's ataxia, a recessive disorder where we're replacing the missing gene called frataxin. A Huntington's program, again, a knockdown program where we're knocking down the gene for Huntington's disease. a gene called HTT, and the final program is for spinal muscular atrophy, again, another recessive disorder. All of these require delivery to either the brain through intracerebral injections or non-invasively intrathecally through the cerebral spinal fluid. All of that needs to be optimized and is being optimized as we advance these programs.
Great. David.
Hi, so I'm David Venables. I'm a CEO of Simpromix, so the European representative here, based in Scotland in the UK. We are, again, different in that we are a synthetic biology company. So we use bioinformatic, data mining, synthetic biology principles to create totally unique synthetic promoters. And we customize those synthetic promoters for very precise and specific purposes. Within the gene therapy area, our focus is on creating tissue-specific promoters, so that the gene of interest is only active in the cell in which you want it to be active, at the strength of expression that you want it to be active at. And so typically, we work to design criteria. We're a platform company. We're not developing our own products, but we're collaborating with cell and gene therapy companies to create synthetic promoters to their design criteria. And typically, those are related to tissue or cell type of indication, to activity level that the indication requires, and also to size. So we create synthetic promoters active in the liver at levels of up to 10-fold higher than CMV or LP1, that are around 200 to 250 base pairs in size. And that's just one example of our design criteria that we work to. In the gene therapy area, we're also developing a new manufacturing platform based on inducible promoters that will enable us to start to create stably producer cell lines for AAV, retro, and lenti.
Great. So I thought we'd kind of cover a few topics related to delivery, transfection, transcription, and talk a little bit on each parameter, how you think about optimizing. Steve, we'll start with you because you're in the clinic looking to optimize CNS delivery of viral vectors. Maybe take us through your thought process and you know, in your answer, if you can integrate having to cross that translational gap from animals to humans, right? As you go and you optimize, what is it that you're experimenting with? Which variables are you playing with? And how do you know that you're on track for the goal of a human therapeutic?
Yeah. Okay, thanks, Josh. So I've been doing drug development, as John has, for quite a few years, and one of the failure modes is not being able to adequately determine the dose and the exposure, the tissue exposure. And I can tell you that we've advanced some drugs that, you know, frankly didn't work because they didn't get to the target, and they didn't get to the target in sufficient quantities to work. So we're kind of hell-bent not to make that mistake in gene therapy. And for our Parkinson's program, we inherited a program that we thought was very interesting and made great pharmacological sense. Said another way, it should work if we could administer enough of the gene to the right area of the brain. The earlier version of the program, which was carried out by an academic lab, in fact, one of our founders, Chris Bankowitz, was a solid program, except for the fact that we now know that there was not enough gene delivered. Safety looked pretty good, but there just simply wasn't enough gene delivered to the right brain region. Now, in our case, we can determine that, and it's a bit unusual for gene therapy programs because we can actually image how much vector and exactly where it's being administered in the brain when it's being administered during what's called a stereotactic neurosurgical procedure, and we can visualize where it's going, and we can say, you know, how much did we get? And from the monkey data, from the non-human primate data, we know roughly what we need to achieve. We can also image the gene product that we're delivering, and we do this routinely now 6 months after the surgery versus just before the surgery. And so we can determine exactly how much of that gene product, the enzyme that we're administering, converts the levodopa to dopamine. So these provide some tremendous handles. In addition to looking clinically, are patients getting better? Are their motor symptoms of Parkinson's disease getting better? So we have a lot of tools, and we think these are absolutely critical for us being able to test this important clinical hypothesis and knowing whether, in fact, this is likely to work or not work as we proceed with the program, and allowing us to actually determine the adequate dose. So again, I would just say that Not all programs have this. Some of our other preclinical programs, we don't have a lot of those tools, if you will. But for this program, we're quite excited because we do have those tools. And I would say that when I've looked at gene therapy programs that have failed, you never really know, did you get enough of the gene delivered to the right tissue, to the right cells in the right tissue? And that's a very frustrating place to be, particularly after you've spent a lot of money.
To what extent, I mean, Steve or any of the other panelists, can you think about just a validation of delivery using that PET imaging approach, irrespective of the gene you're trying to deliver, right? And Steve, as an example, you're trying to get whole brain coverage for certain indications.
Right.
Before you get to a gene of interest, can you just play around with the PET imaging on AADC and say, hey, look at that, we can light up the whole brain? Or, and for some of the other panelists, you know, if you're going into the eye, if you're going into the lungs, if you're going into the liver, is there an opportunity to just validate with an imaging different gene to say, hey, we've checked the box on delivery?
Probably not so much in humans, Josh, but and the other thing I would argue is that a lot of gene delivery protocols are based on preclinical studies, mainly in mice and rats, in rodents. They have very small little brains, and it's easy to get genes wherever you want in those brains. So we use a lot of monkeys in our experiments, non-human primates, and unless we're convinced, for example, that we can either deliver enough of the gene in the monkey brain, which is still a lot smaller than our brain, or the monkey spinal cord, in the case of our spinal cord programs, either deliver enough or knock down enough, in the case of, for example, the ALS program, we really won't proceed to the clinic. We fortunately believe that we can deliver enough, at least to the non-human primate, and so then the leap of faith will be what happens when you get to the human primate, us human beings, and hopefully that'll be a reasonably good proxy for what we'll see in the clinic.
So David and John, how do you think about validating?
4D, we think it's critical to do all of our discovery and our validation of delivery in the most relevant models as close to the human as possible. So we do all of our discovery in non-human primates and or in primary human tissue. So we have a great CRO that we work with that we can do the discovery and the gene marker studies for delivery to specific tissues or even cell types in vivo. And then we also have a human organotypic modeling group internally where we can put together the best panel of target cells as well as off-target human cells and look at the characterization of our vectors and compare them head-to-head with the currently available AAV vectors.
Maybe I can add a little bit to that. I mean, I would echo the animal work. I think that's going to be absolutely fundamental. At Intellia, again, which is a gene editing company as opposed to a gene delivery company, it's really a different problem. It's not trying to get the gene to a place, have it reside there and be expressed for a prolonged period of time. What we're trying to do is get the CRISPR-Cas9 system to the tissue or cells of interest and actually change those genes that are there. So from a distribution, pharmacokinetic, just gene expression problem, it's a very different approach. What you need to do is have the protein there briefly. And we select our targets in a way that we can look for proteins that are knocked down or secreted and be readily measured. Going to the CNS, as attractive as it is, would not be the place where we would start with that, our approach. And as I said in the introductory comments, delivery to the liver is robust with lipid nanoparticles. That's been well worked out. You essentially cover all the hepatocytes, and there's a wealth of different targets that can be readily assayed without going through some of the more extreme sorts of technologies.
So we know, or we think we know where the vector's going, and then in terms of getting into the cells for transfection, I guess David, maybe I'll ask you: do you start with the vector and say, well, this is what our vector can do, therefore we've got a certain number of diseases to approach, or do you start with the disease and say, these are the cells we need to hit? How are we going to get these cells? What's the optimal vector?
Yeah, it's a great question. So I think it's the latter, right? I mean, we have to start with an unmet medical need and then engineer vectors that can basically support a vector in a product that treats that. So I think In each case, either with our own programs or with our partners, we identify the specific tissues we want to hit, the specific cells within those tissues in some cases that we want to hit. Retina, for example, it may be photoreceptor specific, or we may want something that's pan-retinal. And then again, we want to define the most clinically optimal and routine method of administration to target that tissue, because again, we want to get away from some of these heroic measures that the current vectors require. So I think once we've defined that, then we can run a selection in non-human primates that gives us a vector that has the features that we're really looking for.
As we're seeing programs translate from preclinical to clinical, to what extent have some of the successes, failures, and the in-betweens, do you think, have they been driven by either delivery or inadequate transfection? And we'll leave transcription to the side, just getting where it needs to go. Are you confident across all the programs that that translation from animals to humans is robust?
Yeah, I think what we can say in terms of translation from lower species up to non-human primates is there's a poor correlation. So we've run discovery work identifying novel vectors that are great in mice, but you go to a non-human primate and they're really suboptimal. So I think it's critical to get as close to the human as possible. And then again, we don't take it for granted that a vector that looks great in non-human primate will translate to the human. That's why we think having a human organotypic tissue program is so important, to then turn around and validate it on human cells ex vivo prior to going into the clinic, because I don't think you can take that for granted.
We take a twist where you can kind of divide the world into doing pharmacology or fixing a broken gene. And with gene editing, we prefer the latter approach, because you have a very strong link between the genotype and the phenotype. And if you can determine exactly what you need to do from an editing point of view, it should be fairly straightforward once you show in a preclinical model that you can actually accomplish the genetic engineering that you're setting out to do. On the pharmacology side, which is asking cells to behave differently, I think there's always going to be yet another level of risk that comes with that, because you're essentially doing a drug experiment at the same time. And so we've tried to keep it very close to the most validated targets we can imagine.
And the genetics, Josh, very, very important. So if you take, for example, two of our programs, the Friedreich's ataxia program, it's a recessive disorder. We know that if you have one normal allele, one normal gene, you don't become ill. So we're assuming that roughly, we need to give about 50%, roughly, of that gene, of that protein that's encoded by the gene back. And we know exactly where the site is. They're dorsal root ganglia sensory neurons. SMA, very, very similar. It's a recessive disorder. Now, it affects motor neurons. So our task is to define which capsid, which promoter, which vector genome gives us the best transduction of motor neurons, in the case of SMA or ALS, up and down the cord in a monkey. We then take a bit of a leap of faith, because we don't have a lot of human data yet to know how good of a proxy monkeys are to humans. I think that data will come in time. But if we can get 50, 60, 70% of motor neurons expressing our gene, we think that should be pretty good given the genetics of those disorders.
I think, I was going to say, I think you make a very important point that this is a multi-component system, and each part of those components need to be assessed in a valid system, ideally in vivo system, whether that's the capsid, the expression cassette, the promoter, and the gene of interest. And this is one of the issues that we've been tackling with how do we take that complete system in vivo and understand what's the best promoter within that context. And the approach and the solution we've come up with is actually not to look at a protein expression or GFP or luciferase or any of these, but to actually use a bioinformatics approach and actually barcode each element, because we're screening libraries of tens and hundreds of thousands of elements. And so by dosing all of those elements into an in vivo model and then using barcoding to recover actives and optimize the system based on that is proved to be really powerful.
Maybe staying on the topic of transcription, David, so if you figured out delivery and you figured out transfection and the plasmid's there, what are the blocks that prevent adequate transcription, protein production, and how do you optimize and evaluate with various promoters to turn on, to tune in, to dial in the appropriate level of expression?
Yeah, so I mean, the classical approach is to be either to use viral promoters or endogenous promoters. Endogenous promoters give you specificity, but the size is so big that they exceed the packaging capacity of AAV. Or you use a viral promoter, which maybe gives you the strength of expression, but the cell is programmed to silence and shut that down. The way we do it is basically by mining the transcription elements of the cell line itself. So if we, again, take liver as an example, if we understand all of the genes in the liver that are upregulated to make that cell a liver cell, and from each of those genes having identified those genes, mine all of the transcriptional control elements associated with those promoters, and then use those to construct a novel synthetic promoter, essentially you're harnessing the natural machinery of the cell to create the specific expression and to maintain that. If those elements are related to housekeeping genes, then there's no selection pressure to silence those because that's the death of the cell. And so we're looking to basically harness the biology of the system in a synthetic biology tools-based way to get optimal transcription.
I mean, I agree with David. The promoter is very, very important for a number of reasons. One is you want to drive the right level of expression. Too much is not necessarily good, particularly, as I said earlier, in a recessive disorder. You probably don't need a lot for some of these disorders. But also driving expression in the right tissues is important. So when you inject a vector in the cerebrospinal fluid intrathecally, believe it or not, a lot of that vector will get to the liver. And one of the things we're optimizing for is not just do we get expression in the spinal cord or the DRGs, which are in the spinal canal, but how much expression do we get in the liver itself? And we've actually found a few capsids that are pretty good at the spinal cord, but are much, much better at getting that gene expressed where we don't want it, in the liver. So obviously picking the right capsids, designing the right capsids, and then incorporating them in the right vectors with the right promoters. So we have a promoter that we use that works for a long period of time in nerve cells, in neurons, but actually shuts down in liver. We like that promoter because we don't want these genes, several of the ones that we're delivering to the brain, to actually be turned on too much and too high in the liver.
Do we have a sense of the fold difference that a promoter can make in terms of expression?
Oh, it's huge.
Huge.
It's huge.
Yes.
I mean, hundreds of fold in some cases, and the difference between, a CBA or CMV promoter, some of the viral promoters that we and others use, it can be very significant. And interesting enough, the promoters for the natural genes can be very, very low. Now, that doesn't necessarily mean bad. It just simply means very different. And so you need to understand the exposure that you need to correct the disease.
John, can I draw a distinction? Because I think it'd be helpful for the audience. Again, with gene editing, what you're not doing is delivering a transgene and trying to turn it on in a cell. What you're trying to do is take a template that will direct an enzyme to a specific gene and arrange that in a desired fashion so that the native promoter elements are preserved and the cell behaves as you would have expected to in a normal state. So promoters are really not a part of how we think about things. I mean, outside, if you take cells outside the body, you can deliver with ribonucleoproteins, the CRISPR-Cas9 system, change those cells, in situ and put them back after you characterize them. Or in the liver, which again is the area where we're focusing on right now, you can take RNA and deliver it directly and avoid going through this sort of transduction step.
So,
Just one final comment, the importance of the promoter. I think it depends on what you're trying to achieve with your therapeutic. If you have a vector that hits a limited number of cells, say in the liver, but you want to produce a high systemic concentration, the promoter is going to be critical. But if it's a disease where you need to hit as large a proportion of cells in the target organ as possible, then I think the vector is going to be critical to overcome that. You can't overcome that just with a great promoter. So they're both important, but I think it depends on the disease you're going after.
how do we think then? I mean, because we've got variability of the vector, variability of the promoter, certainly tissue to tissue. What about patient to patient? And are there any clues as to what we might expect? And how do you think about designing, optimizing with that, extra variable?
Well, I think it's a very important point, and it gets back to what I called sort of drug discovery 101, maybe 201, and that is you need to define the therapeutic index for each of these vectors as we go into the clinic. Obviously, we do this in pre-IND, preclinical studies, because there is going to be variability from patient to patient, just like there is for almost any drug that you would administer, whether it be small molecules or protein antibodies or whatever. And so one of the things that we're doing in each of our programs is defining, exactly how much gene has to be delivered. And again, we use non-human primates primarily to give us a, quote, a beneficial effect, and we can glean that also from the transgenic mouse data that we have. But most importantly, how much can we deliver before we see any toxicity? And that's really key, because if that therapeutic index is very narrow, you may see problems when you get to the clinic with respect to inter-individual differences in exposure to the right tissue. So you want to ideally pick things where you've got something that's pretty broad, okay? Because when you go in the clinic, it's hard to control a 1 or 2 or even a 3-fold difference, frankly, in how much exposure you're going to get, how much transduction you're going to get.
Yeah, sorry, I'd say we're trying to address that in a couple of ways. One is that one of the reasons there's a lot of variability in dosing with gene therapy today is because of the routes of administration that people are forced to use. So subretinal administration, if you think about it, you're raising a bleb behind the retina, you're detaching the retina, you have no idea what kind of shape or exposure you're going to get there. It's very difficult to control. Obviously, an intravitreal injection is much easier. Likewise with the brain, if you're doing multiple intracranial, craniotomy and multiple injection sites, you really have a difficult time knowing exactly where that vector is going to go. Whereas if you deliver by intrathecal administration, for example, the CSF, you should be able to get a much broader, consistent delivery across the entire brain. So I think that's one approach we're taking is really optimizing for routine delivery methods. And the second is, again, to try to evade the pre-existing immunity population, because those pre-existing antibodies will affect the delivery efficacy from patient to patient, and that's something we're trying to overcome with engineering.
Got it. David, did you have?
Yeah, I was going to say, patient-to-patient variability in terms of the dose they see and the dose that actually transduces into the cells. Obviously, there's so many biological systems at play there that result in that variability. So one of the things we're very excited about is our ability to create inducible promoters. And so there will be some diseases where it's relevant to be able to create a small molecule-induced promoter, where essentially the dose that the patient sees of the gene therapy is defined by the dose of the small molecule that they're given post-induction with the vector, rather than the dose that they see of the vector itself. So I think that's an exciting new element of the toolbox that we're creating.
John, as you, for what you're going to say, also, because what I want to get to is what's unique, I think, about your approach for delivering CRISPR, if you're going to use mRNA or LNPs, is you can repeat administer. And how does that impact or inform your own development strategy as we're talking about some of the variables that you can kind of use repeat dosing to titrate?
That was the point I was going to make, but thank you. I think it's very helpful, actually, because particularly if you're looking for knocking down something that you can measure or building in something that you can measure, you should, in theory, be able to walk up to the point where you want to be. And this is work we're doing on a preclinical basis. We don't have human data yet to verify that, but I think it comes back to this notion of immunogenicity. And if you can truly avoid the immune response, I think that gives you a tremendous opportunity to not have to get it right exactly the first time. And this notion of finding what might be the right dose for an individual patient, I think, is very much enabled as a result of that.
Yes.
So with that, David and Steve, let's come back to AAV, which has the advantage of being a one-time administration, but the disadvantage of being a one-time administration. And David, you've got an interesting toolkit of multiple different kinds of AAVs, but you don't necessarily need to be confined to AAV. You can look at other delivery approaches. How are you kind of thinking about the evolution of the platform, either within AAV or beyond AAV?
Well, I think just starting with AAV, just sticking with AAV for a minute, I think we have the capability of not only evolving for antibody evasion to antibodies that are present in the population at baseline, but also to develop next generation vectors that do not cross immunologically in terms of the antibodies that they generate with the first generation that we use. So we do believe we have an ability to have a life cycle management play where if we do need to come back in the future and redose, we can do it with a vector that does not cross-react with the antibodies generated from the first. It's a little bit like, in cancer with small molecule tyrosine kinases. When you're developing the first vector, you're already developing the next generation for the resistance that develops. And that's the sort of approach we're going to take, is have a next generation vector in hand already that does not cross-resist.
Hey, Josh, just a couple more comments. So the problem of pre-existing immunity is very important. CNS has one advantage in that it is behind the blood-brain barrier, so the amount of neutralizing circulating antibody that gets into the brain is greatly diminished. It's exactly 0.1% roughly of what circulates. However, we always are looking for capsids where there's not a problem with pre-existing immunity. All of us are infected with AAV, and there's cross-reactivity to the various clades or serotypes that we get infected with. So we generally try to find capsids where there's not a lot of pre-existing immunity, but with the brain, either intrathecal dosing or intraparenchymal dosing, we start with a big advantage because the titer is going to be greatly reduced of the circulating antibody to begin with. I think some of the key opportunities will reside in picking capsids that actually get across the blood-brain barrier better than there are some that do now. AAV9, for example, gets better across the blood-brain barrier than, let's say, AAV2, just to pick two, particularly in young animals and young people, supposedly. So we're going to try to find, and we have identified some capsids that get across the blood-brain barrier by at least one order of magnitude better. And then even after intrathecal dosing, either lumbar sacral or cisterna magna, that give broad brain distribution. And I think you'll see in the next year or so examples of those kinds of capsids, which will have, different applications in terms of CNS diseases.
So I think each of the companies has a unique toolkit. And David, we'll start with you. And as you think about applying your toolkit and selecting your first shots on goal, your first therapeutic indications, what's most important, as you choose your top 1, 2, or 3 development programs? What is it you're really looking to establish, and how does it match the unique capabilities of 4D?
So, I believe what we can uniquely provide for the field, for both our own products and for our partners, is these novel, optimized, highly proprietary vectors. So when we select an indication, we're not only selecting a great clinical indication, a great market to go after, but we're also looking for the lowest risk, highest speed indication where we can validate the vector. Because we feel once we've validated that vector for delivery to a particular tissue type, it just opens up a tremendous amount of value that we can leverage across other diseases in that same exact tissue. So for example, if we have an intravitreal vector that targets the retina and we do it in a monogenic recessive disease with clear early near-term endpoints that show delivery, we feel that vector's value just went up exponentially, not because of that one disease, but because of all the other diseases we can also treat. So that's really what we look at, is what are the indications where we can get very rapid, low-risk validation of that particular vector.
And can you talk about the iterative process? Let's say you do go into the clinical setting and it's not quite lining up with your initial objectives. Do you have a process or a conception for, okay, well, that was close or not quite there. What do we do now?
Yeah, I think it's always important to backups for sure. And one of the nice things about the directed vector evolution is that when we complete a discovery process, we don't end up with one vector. We end up with a series of families of vectors, actually. So typically we'll get anywhere from 3 to 5 families, each of which represents a certain capsid solution to the problem. And within each of those families, there may be 2 or 3 lead vectors. So we really have a pool of vectors to choose from. So when we bring a lead vector forward into the clinic and choose it as the lead, we will absolutely have 2 or 3 backups right behind it that have different features that might be advantageous based on what we see in the clinic with the first vector.
And John, how do you wind up focused on the liver, and within all the choices, how do you choose the best starting point for Intellia?
You know, we went through a process similar. I mean, it's an overlapping Venn diagram thing where, you know, what's the nature of the problem you want to solve when you get there, and then how are you going to get there? And it's that design space that determines where you go. And in the case of Intellia, with the relationship we struck with Novartis, we gained access to their very deep lipid nanoparticle library that they had worked on for many, many years. So it's a real advantage for us to get going and not start from scratch. And then when we look at the liver, we view it very much as a target-rich environment. I mean, there are literally hundreds of different disorders one can go after, some of them common, some of them very rare. And so what we've tried to do is stratify them based on the nature of the edit necessary to correct them when you get there. And then what is correction? You know, it's similar to some of the points that have been made here. Some of these disorders are enzymatic in nature, and a simple change in the order of 1% to 2% of native activity will restore essentially a normal phenotype. Others are much more complicated than that, but we've essentially stratified literally a couple hundred targets there. So, you know, your point, David, of once you get into a room, so to speak, once you walk through that first door, ideally you'd like to be in a place where there are many, many targets to continue to pursue, and I think that's what we've achieved.
And Steve, with the Parkinson's program, has everything gone exactly according to plan? And if not, how do you react? Like what's the process you have in place to make sure that, you know, where you want things to go are lining up with where things are going?
Yeah. Well, of course, nothing ever goes exactly as planned, right? But, you know, in our case, we very systematically have embarked on a dose ranging study because this is an irreversible one-time treatment. Obviously, you don't want to give too much of the vector. You want to go up thoughtfully and safely on the dose response curve. And when we did the first 5 patients at the lower dose in this phase 1b study, we had to be very objective. You know, we saw good safety. We saw about 20% coverage, but quite bluntly, we didn't see a clinical effect that really got us excited. You know, probably if this was an academic study and they never knew how much coverage they were getting, we have built that into the trial, or how much enzyme they were delivering, you might have said time out, probably published a paper, declared victory, and moved on, right? We decided and said, look, that data is interesting and important, and it says that this dose is not the right dose. Go back to the monkey data, and sure enough, the monkey data was very informative. You're not going to see much until you get to 35%-ish, roughly coverage of this area. And the concentration of the viral vector was key too, because that determined how many cells in that region of the putamen were actually producing the protein. So we just, you know, filed a protocol amendment. The FDA said, we agree, go up, and we doubled the dose. And it was really the very first patient where we doubled the dose and got the coverage that we think is reasonable, that we started seeing these clinical effects. Now, here's where it gets interesting. Do we optimize even further? Well, we want to get a few more patients under our belt, see what they look like, but there's actually room in our case to actually increase the dose by another 6-fold with the current formulated vector that we have. I'm not saying we're going to go there, but it's possible. So we want to do this very thoughtfully, because we think that if we get something clinical endpoints like we did in this very first patient of the higher dose, you know, we'll have a potential treatment for patients, and that's the goal.
And David, optimizing promoters, you can go anywhere. So where do you start?
Yeah, no, we can go anywhere, and that's what makes it really exciting. I mean, we've done work in gene therapy, anything from gene therapy through to plants. But obviously for us, where my background is cell and gene therapy, that's somewhere where we see huge opportunity. We're really excited by that. We're very excited by the collaborations we've got in place now with AGTC, Avalanche, Unicure, and a couple of other pharma companies that we're working with. We've got in total about 9 cell and gene therapy programs underway. So we're looking forward to seeing non-primate data this year. So it's really exciting. But I think for the next stage for us, it's starting to mine the inducible components much more. Very interested to hear the mention this morning about armed CAR T cells, for example, with 2 to 3 genes. Now, talking to those guys, the reason why it's not 5, 6, or 7 genes is often that the packaging capacity is just too small because the promoters are too big. If we're down below 200 base pairs of promoter, you can start to think about arming multiple genes into CAR Ts, each of which is induced by a different environmental condition. And so that, for us, this year, that's a very exciting area for us to get into next.
Let's see if there are any questions in the audience. And because I can keep going, and I will. So David, hopefully we can come back in a year because this is a topic that's going to persist for years and years, optimizing the approaches. So where do you hope to be when we come back and talk in a year from now?
Well, we've currently got 15 different discovery programs ongoing targeting specific cells and tissues in the body. So we expect to have those completed in the next year. We also would like to have completed in vivo and ex vivo characterization of our lead vector families from each of these selections. Again, important component of that will be comparing them head to head with the currently available vectors and hopefully we believe showing superiority. So I think coming in with a completed those 15 discovery programs and having validated at least the first half of those will be important. And then we should be well on our way to completing the IND enabling studies for our first 2 products in choroideremia and cystic fibrosis.
Great. John, where are you guys in a year?
In a year? Okay. Back here at this meeting, I guess. You know, if you start from any gene, anyhow, anywhere, we've seen what, you know, the potential of a platform like CRISPR-Cas9 provides. I mean, the opportunities are limitless. It's not going to be ideal for every single problem, but I think it can address many that have been very difficult to go after. As we build our company, we're thinking about the technologies we need to aggregate so that we can, you know, capture as much as possible of that potential. We've talked a lot about lipid nanoparticles and what that does for us in a very target-rich tissue like the liver. We look for partners who can help us build that technology out so that we can reach other tissues that hold the same promise. And we spend a lot of time pursuing that. I don't think we'll be in the clinic a year from now. I wish I could say that, but I believe that our lead programs, which we haven't even announced, will be well on their way to that pre-IND sort of phase of their work.
Will you have announced the programs a year from now?
Absolutely.
Perfect. Steve?
So for us, Josh, there are kind of 3 areas that we look forward to. One is the readout of our clinical data from this phase 1b study that'll occur, top line readout Q4 of this year. We'll have the 10 patients dosed, we'll have the pre-baseline biomarkers, we'll have the 6-month biomarkers plus the clinical endpoints, and we'll be able to get a sense of, is this working? Have we picked roughly the right dose? Do we need to go up on the dose? Those are the kinds of parameters. We'll have that data, fortunately, we believe, by fourth quarter. We'll have hopefully selected the candidates to enter pre-IND studies for the next 2 programs. This would be the ALS SOD1 program and the Friedreich's program. It takes about 11 to 12 months to do the pre-IND work in order to get an IND submitted and to start dosing patients. So hopefully we'll get the R to D transition occurring towards the end of this year so that we could be in the clinic with one or both programs before the end of next year. It's a big thing. And then the third element, and we made a lot of progress in this already, is we've begun the GMP manufacturing activities for the Parkinson's product at our Fall River manufacturing site, the site that we're developing with Mass Biologics. So we're using this baculovirus SF9 system, which allows us to scale up and produce both research grade vector, but also GMP grade vector, importantly. And we're using, obviously, our Parkinson's program as the lead program to get that manufacturing process up and running for the company. That'll all be done within the next 6-8 months or so.
Great. David?
So in 12 months, then we will have completed the non-human primate validation of our promoters, and we're optimistic of some good data there. We'll also have built out our inducible promoter franchise, if you like, specifically in the area of CAR-T and also control of CRISPR. And we'll have developed our next generation vector manufacturing platform for using inducible promoters to optimize AAV vector yields.
Great.
Well, and in our spare time, I have no idea what we'll be doing.
Exactly. Well, a terrific discussion. I think just the tip of the iceberg for what we're hopefully going to see. Since Piper is one of the sponsors of the IRM event, I hope I get to plug our own gene therapy event, Genomeric Symposium, May 17th in New York, where we're going to hopefully elaborate on a lot of these topics in terms of pushing the field, the platforms for gene therapy, gene editing, CAR-T forward, and where it's all going to go from here. So for our panelists, thank you so much. Best of luck for the year to come, and I think in a year from now, my prediction is that we'll have 4 publicly traded companies. Thanks everyone for listening. We're going to get started in just a couple of minutes with the next company presentation. Thank you.
Thank you.